to talk about lesson 3 with only a small review of lipids at the very beginning of class. So where we'll start today then is our third of 4 biomolecules, which is the protein. So we've now talked about carbs, right, aka sugars. We've talked about lipids, which are like oils and fats. And so now we're going to talk about proteins, which I will continue to joke for pretty much the entirety of your semester with me until you're horrifically sick of it, that it is our favorite biomolecule. And you'll see why I continue to say this, right? Pretty much by the time we're done talking about this, but certainly several units in, when they kind of continue to keep cropping up. English. All right, so here's my orange box again. Right, so remember that our key here is the same that it's always been. Right, we still want to have a good idea of what is the purpose of a protein, and besides, again, being delicious. And, right, how would I recognize or identify the structure of a protein? Okay. Now, again, this is going to become a little bit more and more simple for us, okay, comparatively speaking, because now we've seen 2 different biomolecules that we can kind of reflect back on as we go, thinking about, okay, well, these are the structures I have seen. Okay, what does a protein have or not have? Okay, what functions do proteins do or not do? Okay, so one of the main reasons that we talk about proteins being our favorite biomolecule is because they are extraordinarily versatile. Okay, a lot of the major stuff that we're going to talk about this semester are going to have proteins at their core. Okay, so I have some very general functions up here. Okay, and we're going to encounter a lot of these throughout the core of class. Okay, so for example, proteins can make enzymes. Okay, we'll see those often have these ASE endings. Okay, so enzymes are things that break down, okay, other compounds. So for example, the enzyme lactase, right, ACE ending, exists to help break down milk sugars, right, lactose. Okay. In addition, right, proteins can be storage or structure. Okay, we eat proteins, okay, and that becomes part of our body storage. They become stored in our muscle. They help build muscle. Okay, and we'll see very lightly when we're talking about cell transport, okay, that they also help to both protect the cell and then move information around. Okay, so they're really, as we'll find, some in this semester, and then as you take some later classes, there's really nothing that proteins don't have their little fingers in. I feel okay so far? And this is very general. Okay, so what does a protein look like? Okay, so proteins are made up of amino acids. So proteins are like the really big compound, right, the final product. And so we're going to talk about how do we build up to making that final product. So the subunits, kind of like the Legos that make a Lego castle. Okay, so the Legos here are the amino acids. Okay, and you might have heard about amino acids before. Okay, so of amino acids, okay, 20 occur naturally. Okay, you can find them in nature. Of those 20, you need 9 to survive. Okay, your body uses the materials within those amino acids as core structural and nutritional pieces within your cells. Without them, your body will cease to exist. Okay. If you're a big sci-fi or fantasy fan, like the core of the Jurassic Park series, it's actually based on this concept, right? If you're real big into the actual story. So the dinosaurs in the Jurassic Park series are bred without lysine, right? It's a lysine deficiency, that's what they mean. Lysine is one of the 9 naturally occurring essential amino acids. All right, so without that amino acid, the dinosaurs theoretically are supposed to die. It's their fail-safe. So they have to provide the dinosaurs that lysine in their food. If they stop feeding the dinosaurs, theoretically they would die. That's why they're not supposed to escape, hence plot the movie. It horrifically fails, whatever. But that's where the idea comes from. And that's what they mean when they say that. So you can go back and watch like the original movie or the books. They're way better. That's what they're talking about when they mean that. So what exactly are these Legos? Okay, these pieces that make up a protein. How would we identify one if we saw one? Okay, so let's look at the image I have over here. So first let's focus on the blue box. Okay, we've seen the blue box before. Okay, remember that means other things. Okay, we're actually going to come back to that in a hot second. Okay, but for now, we're just going to recognize it for what it is, right? Lazy chemistry labeling. Okay, just other, something else, right? So instead, let's focus on my red boxes. Okay, the red box on my right-hand side is familiar to us. Okay, we've seen this group before. All right, so here's a carboxyl group, right? Here's the carb part. Okay, and then the oxyl, right, from hydroxyl. Okay, so this is something we can look for and see, and this will help us know it's an amino acid, but this isn't the only compound so far that's had one of these, right? What other biomolecule also has carboxyl groups? Lipids. Okay, so this is useful. Knowing amino acids have carboxyl groups, so we want to be careful, right? Because it's not exclusive, right? Because our lipids, if you flip back and look, also have the carboxyl group. Okay, so carbohydrates do not. What do carbohydrates have instead? Carbonyl, good. Okay, which is very similar. Remember we said we want to kind of keep track of this stuff. All right, so carbs, or carbohydrates, right, have the carbonyl group, right, which is the C to the double bonded O with the H hanging off at the end. Okay, so far it's the only one we've seen that's had a carbonyl group. So that's a very useful tool. Both our lipids and our amino acids have had the carboxyl group. Still useful, okay, but just useful while being careful. So we need other tools that can help us recognize these things. Lipids had other things we could look for. For example, those long hydrocarbon deserts. So for amino acids, we're going to look for this other red box, the one I have on the left side here. Okay, so where it's getting its namesake. So here we have what's called the amino group. Okay, so amino acid, we have an amino group. Okay, so this one looks a lot different than anything we've seen so far. Okay, this isn't just some combination of carbons and hydrogens now. We have a brand new element to play with. Nitrogen. Okay, so this is very simplistic, right? It's just a nitrogen with two hydrogens sticking off it. But it's very easy to recognize, right? Because so far, at least, it's the only compound we've seen that's had nitrogen in it. Okay. So I would want to check first for the amino and then the carboxyl if I was looking for these things. So it's like having two arms, but they're very, very different sizes and shapes. This will become important. So we're going to have my left arm's always going to be an amino group. Okay, my right arm's going to always be that kind of longer carboxyl group. A little asymmetrical. Okay, and that's helpful too for recognizing it. Okay, any questions so far? Okay, so for one of the first times ever, we are going to focus in a little bit on this R group. Okay, so remember I said that there were a whole bunch, right? 20 different amino acids that make up my proteins, right? Different kinds of Lego pieces. And if I just look at this as is, okay, all of these pieces here, everything in the red, okay, and that middle piece below my blue box is always going to be exactly the same. Okay, none of that can change. That's what makes it an amino acid. So if I'm going to get 20 different ones, okay, there's only one place that we can get that kind of diversity. And that's in this R group, the other part. Okay, so what actually happens in our R group here matters, as we're going to find, for building our final protein. So we have a lot of diversity in that R group, and that's going to give us these 20 different kinds of amino acids. Okay, and we'll recognize the types of things that are going to come out of there, right? We'll get some polar groups. What does it mean to be polar? Have lots of oxygen. Good. Okay, or non-polar groups, meaning we don't have any oxygen, right? I'll elbow this a lot. Okay, we also might be charged or not charged. Okay, literally have a positive or negative charge to it. Okay, and the presence and absence of these things are going to give us some diversity. So let's take a look at what these look like. Use the word look a lot in one sentence. Okay, so at no point will you ever have to have these memorized for this class, right? So the screen is not up here, so we have to memorize what glycine looks like or any of that business, right? So we're not panicking as we look at this screen. This is merely an example so we understand, okay, what are the R groups. So my R groups or the other groups are all colored for you here, right? They're always the things in the block of colors. Okay, so here are the things that we sort of recognize. So in my top set, everything that's in green are all non-polar, right? Let's call it my green. So remember this oops, sorry about that. Non-polar, right? Ooh, that kind of looks like an N. Fix that. Okay. Okay, doesn't play well, right? No play with other things. Okay, so remember here we would expect not to see a lot of oxygen. So if we take just isoleucine, for example, because it's close, okay, in my green box, which is where that R group, that other group is, I don't see any oxygen there. All carbons and hydrogens, just like we saw when we did lipids, which is a really good example of a non-polar group, right? All right. Same thing for all of these. As I look at them, okay, remember for these that are kind of just drawn on there, what do the bends represent? Carbons, good, right? So these all carbon chains in there. Okay, so none of these have any oxygen in them. Some of them have weird stuff, okay, but it's never oxygen. Now, if I pop down to my purple section, okay, these are polar. Remember, polar things play well, right, because they have at least some oxygen or charged ion in them. Okay, cytosine is really weird. We're just not going to deal with the chemistry behind that one. Okay. So this fulfills our expectations of what we're kind of used to seeing. Okay, carbs have kind of prepped us for this sort of business. Okay, so the only thing that's super new is our bottom box here, right? Some charged ions, okay? But these are also more or less intuitive, right? If we look here, I can literally circle parts that are quite literally negatively charged or like a scavenger hunt here, positively charged. So it's sort of like ions and magnets, right? There's a positive or a negative version of these. So what the R group is on our amino acids is going to indicate to us how these are eventually going to interact. And this is going to be important, remember, as we think about each amino acid being a kind of Lego piece. And we're going to eventually put our Lego pieces together to build something. Okay, so in this case, right, only certain Lego pieces will fit together. We fit together nicely. Okay, does everybody understand what we're looking at when we see this big diagram? Right, and it's not horrifically intimidating or overwhelming to look at. So we really want to focus on the key pieces here. Okay, so if we want to link our amino acids together, how does that work? Okay, so same boring process we've been doing the whole time. Okay, so we're still going to use dehydration synthesis to put them together and hydrolysis to break them apart. Okay, so again, these look different, so it's going to feel kind of intimidating and overwhelming at first. Okay, so all we need to do is find the things we recognize, okay, to allow us to do that same process we've been doing from the start. So let's do just that. I'm going to assign amino acid 1. Let's see, who am I taking the arm off of here? We're going to make that Carl. We always take the arm off of Carl here. All right, so we've got Carl and Janet. Boy, these two really need to get it together. Okay. Now, if I look at Carl, okay, I have some familiar compounds to start with. Okay, remember my goal when I'm dehydrating, right, is I want to pull out, dehydrate out a water molecule. So I have H2O that I want to get, right? That's my goal. I know from my process, some way or another, that is what has to happen here. Okay, so let's start with Carl. Okay, Carl has some familiar compounds. Okay, he's got a carboxyl group, so let's start with that. We've used carboxyl groups before, right? So when we did lipids, remember we used carboxyl groups in dehydration synthesis? So let's do that again. So remember when we did the carboxyl groups, okay, this has 2 of the pieces we need to make our water. So we can take those. All right, so I've got my first H here, and check that off. Okay, so Carl loses his mitten. Okay, and I have a nice little oxygen here. So I can check that off. So Carl loses his arm or hand. Okay, sorry, Carl. All right, so we're doing the same thing we've done so far, right? We've taken Carl's arm. And we've taken the oxygen and one of the hydrogens that we need to make water. It's a handy little checklist we've got going. Okay, but we're still short, right? We need one more hydrogen in order to do our dehydration. Before we do anything else, we have to take one more thing, right? So let's go over and look at Janet. So Janet, right, we have a new side group to be working with, this amino group. Okay, and it looks weird. Okay, this nitrogen business is weird. However, okay, my goal is simple. All I need is a hydrogen. Okay, my amino group does have hydrogens. Okay, fine. I'm just going to take one of those then. Okay, that will fulfill my checklist. Okay, so I'm taking one of Janet's mittens here. Okay, by doing those 3 things, right, we're sticking to our checklist. That's the thing we're familiar with. We've made that checklist for all of our dehydration synthesis up to this point. Okay, so now I've made water. Yay! Good so far? Everybody see what I've done when I took those pieces? That's all we've done. That kind of feel familiar? Okay. This is a good way to kind of tackle these, right? I know I need to make this stuff. So where is the best place to take them? I know I need to take some from Carl and some from Janet. All right, so let's switch colors. Okay, so now, right, Janet and Carl are both missing hands and mittens and all sorts of stuff, and they're ready to hold hands. Right, that was the whole goal. Okay, so Carl, right, having none of this left, is going to have to come out from this carbon. That's all that's left. He's got nowhere else to go. Okay, same thing for Janet. Okay, we took this hydrogen away from the nitrogen so we can come out from our nitrogen now. It's kind of a weird bond, but it's what we have set up, so we're going to roll with it. Okay, so here's that bond there that we set up. Okay, we connected Carl's carbon to Janet's nitrogen, and that's our handhold. It's a little harder to recognize as a handhold, but we have connected them together. My dotted line shows up. Okay, does everybody see why we connected the carbon from Carl and the nitrogen from Janet? Jasmine, am I recording? gracious, thank you. We are really far in now. Okay, so just like before, right, when we did carbs, right, that simple carbon to oxygen to carbon to bond, what kind of bond was that called? Okay, in lipids, we ended up doing something a little different, right? Because we were using that carboxyl group. Oh, that's definitely an O. Okay, what was that called? An ester bond. Good. Okay, so, right, we changed the name in lipids, right, because we were using kind of a funky connection. Okay, same thing here, right? This is a funky connection. So now in proteins, we have our, whoop, I didn't leave myself even remotely enough space, did I? We still have that carboxyl start, but instead of going to an O, we're going to an N, right? So now we have to have a new name. This is called a peptide bond. And so the result looks a little weird, right? That carbon to nitrogen connection, making that kind of peptide look. But everything we did, right, using our checklist, was exactly the same as what we've done since we first learned this with carbohydrates. Okay, any questions about what we did here, creating our very first peptide bond? Okay. So as I mentioned, okay, our goal is to make a protein, right? And the protein's big, and we're starting with small things, right? The amino acids, like little Legos. Now, our big protein, remember, has a large diversity of things we can make. Okay? It's like getting a Lego set that has multiple kinds of instructions on the different kinds of things you can do. So the way that you put your Legos together is going to result in a different final product. And in this case, right, the final product is going to go do something really important. So what we're going to look at next is how can you put together our Lego sets in such a way that allows for such a large amount of functional variety in our proteins, right, when you really only have like a few different kinds of pieces to work with. Right? I mean, I can put pieces in any order, but in the end, you still have to make a castle, right? Still has to do something. It can't just be a blob of stuff on the floor. So let's look at how we actually make that big castle at the end. Okay, so we call these levels of organization. Organization kind of referring to the building or shaping of. So we're going to start at primary, and we're going to go all the way up and see how do we make really big cellular proteins. Okay, so the very first step that we need to take in building our shape, okay, or our primary level of organization is pretty much what we just did. what we just learned to do using dehydration synthesis. Okay, so we can call this, stop, there we go, the amino acid sequence, right? So literally just take all of our different amino acids, okay, and link them together by these peptide bonds, right? So we're using that dehydration synthesis process. So, wow, okay, yep, good job. There we go. So we pretty much just do, okay, that previous slide anywhere from 2 to 500,000 times, right, depending on how long we need our chain to be. All right, so this isn't so bad. The thing we want to keep in mind is, you notice I make a big deal about this being linear or flat. Okay, and so we're really going to talk about flat structures versus three-dimensional structures, and this is going to be a really important stage. Okay, so as we've done this, okay, we're pretty much just linking these up on a flat plane next to each other. Okay, so just going to make, kind of imagine like linking a chain and laying that chain flat on a table. Okay, the chain's not going to kink or fold or pull up in the air, so you're just laying it nice and long along a table. Right, you can add as many links as you want to your chain, okay, but it's still just going to lay flat. Okay, and that's sort of what we have imagined here, right, is a long chain or beaded pearl set, whatever it is you want to imagine your amino acids as. Okay, just all being hooked together, one after the other, using that dehydration synthesis process, creating those peptide bonds, right, those carboxyl to nitrogen bonds. Okay, so primary level of organization is intuitive. This is pretty much what all of the other biomolecules stop at. Like, yes, okay, we made a thing. Okay, but proteins have much more diversity than that, so they need to keep going from here. So after we start linking stuff up, we're going to start twisting and folding our product to give it a useful shape. And the shapes that we make are going to ultimately decide what the function of our compound can be, what our capsule can do. Okay. So step 2, right, our secondary level of organization. Okay, we have a combination of 2 different shapes that we can make. Okay, I like to call this the leftovers stage. You'll see why in a hot second. Okay, so we're going to make what are called alpha helices. That's our left image here. Looks a bit like a spiral. Okay, or beta pleated sheets, which are these kinks you see on the right-hand side. Kind of looks like when you fold a fan out of paper. Okay, these are created due to hydrogen bonding. Okay, so what's a hydrogen bond and how is this happening? So this is where the leftover concept comes from. So if we think about our peptide bonds, this is a peptide bond, right? And then there's like more crap to the left and the right. So remember when we pulled stuff off of our carboxyl group, we took the OH, sorry Carl, but we left the double bonded O. We didn't take that away, we left that there. Okay, when we took Janet's 1 hydrogen, right? We took her mitten. She had 2. Okay, so she has 1 of those left. So these are our leftovers. Okay. Oxygen and hydrogen are highly attracted to each other. This has a lot to do with chemistry stuff. But in the end, remember, we said that oxygen's like this selfish roommate. Right? So oxygen's always a little bit negative. Hydrogen's always a little bit positive. Okay? The key, or what I care that you remember, is they really, just really want to hang out hardcore. So we've got a whole bunch of these leftovers just chillaxing in our long primary chain, right? So anytime an oxygen and a hydrogen get very close together, they want to hook up. It's like a bad Tinder date. So this is what we mean by hydrogen bonding, okay? When oxygens and hydrogens get very close together, right, they pull each other very close and become bonded. And so that's what we see here. Okay, a whole bunch of places where those leftovers, okay, have gotten very close to each other and are pulling, bonded, or pulling on each other. And they're creating these shapes that we have. So here we have a hydrogen bonded shape. Okay, any individual chain of our peptides should have both, usually in multiple places, alpha helices and beta pleated sheets. Okay, which is extremely hard to do in my demonstration, so I'm only going to show the beta pleated sheet because it's much easier to do. Okay. So our hydrogen bonds have created these very organized repeating patterns. Okay, now, here's the thing to keep in mind. Stop, unfold, but when we're doing this, okay, we are still 2-dimensional here. Now we've created some folds and kinks, but when we think about something being 3-dimensional, okay, I really want to picture like Lego castle, right? Something that's tall and wide and you can walk in, right? So it's got roundness and depth to it. Okay. Now we've kinked up a little, so we imagine, right, our Legos maybe have multiple layers of Legos, okay, but it's not really anything useful. Okay, so here is why this is really not 3-dimensional, right? So we have, all I've done here is the pleated sheets, right? Please ignore the floppy notebook underneath. But even though we've pleated up, right, this is still quite flat. Okay, it's a little 3D, okay, but this is laying quite flat on the table. It's hardly any bigger, right, any taller, any less 2-dimensional. Can you see this in the camera? Okay, good. Than what we were doing in our primary stage. And it would be the same with the spiral, right? It's just hard to do both a spiral and a beta pleated sheet on the same piece of paper. Okay, so this is still secondary 2D, right? Step 2 is still 2D, 2-dimensional. Okay, it's still very flat. Okay, so this is a much more complicated bonding section. Does everybody see both what the final result is, why that's two-dimensional, and where the hydrogen bonds are coming from? All right. Our tertiary structure then is where what we call side chain, our side chain interactions come in. When we're talking about side chain interactions, what we really mean is the R groups. Okay, so if we think back to that sheet, okay, we know some good rules on how those things interact. So if, for example, I just look at my pink box right now, okay, we see that this is identified as hydrophobic interactions. Okay, we've seen that before. Okay, this is what happened with our lipids. Okay, remember, this is what happens when two non-polar groups get together, right? Remember, they like to tuck their tails in together, right, and hide, right? They don't interact with anybody else, okay, but they interact really well together. Remember, this is how we created our phospholipids. Okay, you'll continue to see some hydrogen bonding. Okay, remember, we have more polar groups. So we have lots of oxygen and hydrogens coming to the party. So we can do more folding that way. Okay, we can do some ionic bonding, okay, which is just literally a positive and a negative coming to hang out, right? Remember, opposites attract. Okay, so there's almost an infinite version of stuff that can come up here. But the key is that diversity of stuff that we saw in those R groups, okay, is going to give us lots of different options, okay, depending on where they are, right, and how close they are together, of how they're going to hook up. Okay, so what amino acids that we put into our original primary chain and where they are, aka how close they are to other amino acids, will help dictate how this bunches up and folds. Okay, but pretty much the key here is if I think about having my rather flat pinched or spiraled protein, is if I have an amino acid here that's nonpolar, get in my fingers, an amino acid here that's nonpolar, right? They're going to be attracted together, so we're going to fold up. Okay, same thing here. Let's fix this so it actually does it. Okay, same thing down here. Maybe I have a hydrogen bond here and a hydrogen bond here, right? So this is gonna, right, pinch up. This looks like I'm crumpling, but this is awful hard to do. Okay, so we're gonna get different pinches and twists. Maybe we're gonna come around. I kind of lost my beta pleated sheet, but that's unintentional. Okay, so in the end, there's someone with better origami skills than me. Okay, what you'll end up with is a structure that is now got depth to it. Okay, so and this is the key, tertiary, third step. And we are officially three-dimensional. Okay? And as soon as we're three-dimensional, we're functional. Right? We're all grown up. Go out and get a job. All right? This can go out and do whatever it's supposed to do. Okay, so if I'm lactase, right, that enzyme, I can go out, chomp, 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 break down that sugar. Yeah, almost looks like an enzyme. Okay, any questions about our tertiary level, right? Our three-dimensional amino acid or protein organization. Okay, we'll go ahead and stop there today then. We'll compare tertiary to quaternary on Friday. Got a giant paper mess up here.
So also, please don't forget my study session tomorrow at